Not that long ago, my dad asked me the very simple question, what about Turkish hair mushrooms? Now, without the context, that sounds like a stupid question, doesn't sound like a sensible question at all. But the context was that he had watched a podcast where one of the guys on the podcast had a dog with cancer, and giving that dog turkey tail mushroom extract actually rolled back the cancer and made it vanish within months. Now, that is a very cool anecdote. It's interesting, but I have to say, I was quite skeptical. Now, I want to call myself a good scientist, right? So I did the only sensible thing, and I perused the literature. I actually looked at, mushrooms in general. Actually, I didn't start out with turkey tail mushrooms specifically, and I found pretty quickly a mushroom that has enormous promise in cancer care and actually has shown very, very positive results. That mushroom was the subject of the last video, and that mushroom is Ganoderma lucidum. In this video, we'll talk about another mushroom that also shows very, very good promise in anti-cancer care, or in anti-cancer treatment, cancer care, which is Cordyceps sinensis. If you don't know who I am, my name is Merlin. I have a bachelor's degree in molecular biotechnology. I'm working on my master's degree, and I'm one of the co-founders of Marchwood, where we try to eradicate cancer as a cause of death. If you want to know more about our approach to cancer and the science behind it, you can go to marchwood.com and just read through our article of libraries there for free. With that out of the way, let's get into the video. Okay, so let's look a bit at the context of looking into Cordyceps sinensis and why it actually was interesting to me upon the first cursory glances at it. Now, what we're going to look at here is the kind of aspects of cancer that make cancer dangerous. So in tumor formation, and that is both benign and malign tumors, so both non-cancerous and cancerous tumors, we see that we have as kind of attributes. So if we have a tumor here, we see one of the attributes is a decrease in apoptosis. Apoptosis is a kind of self-sacrificial cell death that the cell will enact when it is defective or infected, or if it just needs to die to kind of make space for new cells. And it's used a lot in multicellular organisms, which you and I are, to kind of keep a proper balance between dying and growing cells. On the flip side of this, what we see in tumors is an upregulation in growth. So just normal cell growth. In tumors, the cells just divide more rapidly and have lower apoptosis. Some tumors just have better growth and normal apoptosis. Some tumors have lower apoptosis and normal growth. But at the end of the day, if you have a lower amount of death and a higher amount of growth than should be normal in your tissue, then you will kind of develop these tumors, these bulbs of tissue that just grow where they shouldn't. And the interesting thing is that cordycepin, which is one of the compounds in Cordyceps sinensis and also in related Cordyceps and Ophiocordyceps mushrooms, what it actually does is it counteracts this and this. So just in normal tumor formation, it's already helpful because it upregulates apoptosis and it downregulates growth. Now, it doesn't just strictly force a downregulation of growth, but it has an effect on some of the proteins involved in cell cycle, so that the transitions between some of the phases in the cell cycle aren't as easy for the tumor to go through. And likewise, it also just doesn't put a cell into apoptosis, but it kind of shifts the regulatory pathways of apoptosis to make apoptosis easier, if that makes sense. So it doesn't just kill the cell in apoptosis, but it kind of makes apoptosis easier so that these defective cells have an easier time of killing themselves or of being killed by extrinsic. So extrinsic apoptosis, which is caused by immune cells or other cues from the environment to the cell that they should kill themselves. Now, this is all tumors. This is not just cancerous tumors. This is all tumors where this is effective. But if we go and we kind of mature this tumor into a full-blown malign cancer, then we can actually see here that these cancers have two other attributes that we don't want. Namely, cancer is invasive, so we have the attribute of invasion. And cancer is oftentimes, especially very advanced cancers and mature cancers, are oftentimes metastatic, meaning they enter into the blood and then they travel into the blood and then they kind of settle down and build a new colony, so to speak, in a distal tissue, in a like tissue that's removed from the first site of cancer or tumor. And in tumors, we see that obviously both of these, or in cancer tumors, we see that both of these are upregulated. And obviously, normal cells don't invade and normal cells don't metastasize. That's kind of, a exclusive characteristic of cancers. And wouldn't you know it, cordycepin actually lowers or counteracts these, so lowers both invasion and metastasis in certain cancers where we have the data. Now, it's probable that it does that in all cancers, but we have data for bladder and prostate cancer where it lowers the invasion and metastasis of these cancers. And I think that kind of is already enough to make Cordyceps sinensis very, very interesting to cancer patients. But it gets better because Cordyceps extracts or Cordyceps sinensis extracts specifically also improve general immune function and cytotoxic efficacy of the immune system against cancer cells, meaning the immune system is now more active, more vigilant, and is better able at killing cancer cells. I think that just makes it even more interesting, and that was the starting point for me to look into it more deeply. And if you want to read through this all yourself, you can go to the article linked below, where I have all the references, all the citations, and of course, everything, all the background of this explained perhaps in a bit more depth than I can do in this video. Now, I just mentioned that Cordyceps sinensis takes influence on the immune system. What I want to do here is to first look at what cancer does to the immune system and then how Cordyceps kind of counteracts that. So if we take here, let's say just a normal, a good old normal cancer. I don't know how so far cancer is normal. It is horrible, but anyway, we have cancer here. And what we see that cancer does is it promotes the polarization of macrophages into what we call M2 macrophage, or macrophages. These M2 macrophages are a specific subtype of macrophage. Now, macrophage is a kind of immune cell that's always active. We call it part of the innate immune system. Innate because it's always active and it's not adaptive. It's just, it does its thing. And then we have the adaptive immune system, which is largely dependent on lymphocytes. But that's a different discussion. These M2 macrophages, they have a kind of secretory pattern. So they secrete cytokines, or they secrete compounds, molecules that have a immunosuppressive effect. And cancer supports this maturation or polarization of macrophages into M2 type macrophages, which suppress the immune system. Specifically, they suppress T killer cells. So what this also does, this is kind of a direct effect of cancer through acidity in the cancer microenvironment or tumor microenvironment, and also an indirect effect through the upregulation or promotion of M2 macrophage polarization, and that is a depletion of cytotoxic T cells. So cytotoxic T cells are what we call CD8 positive. It's just a surface marker that is available on cytotoxic T cells. But what we see here is that cancer depletes these cytotoxic T cells, and that is bad for us because cytotoxic T cells ordinarily would kill off cancer cells. Now, cancer also has other ways of kind of circumnavigating cytotoxicity. One is, of course, this depletion of T cells, but cancer can also just signal for, or basically ask T cells for asylum and stop them from killing the cancer cell. It does this through various means. I'll call this direct immune evasion. It's not an actual official, I don't think, at least, I've never seen it written, direct immune evasion. But what I mean with this is basically the expression of certain surface markers on the cancer that signal to T cells or other immune cells to leave the cancer alone. And cancer does that readily and a lot. And we don't really want that because it's kind of bad for our purpose of killing the cancer. And what cancer also does is it promotes the formation or the maturation of T cells into a specific immunosuppressive phenotype or shape. And this is obviously, if you see this immunosuppressive, this is bad. At least in cancer, it's bad. Because we want the immune system to be active against cancer, not suppressed against cancer. Okay, so this is how cancer influences the immune system, both indirectly and directly. Now, what does Cordyceps sinensis do to this? Well, Cordyceps sinensis actually improves the maturation of T cell. So C sinensis improves T cell development. I'm not sure how to graphicalize this. Let's say it downregulates the depletion. So this is no longer as dominant a factor, I would say. It's probably still a factor because none of these things are perfect, as biology is more a game of balances. But Cordyceps sinensis counteracts the depletion of T cells by promoting the maturation of new T cells. And it also delays this. So it also downregulates the maturation of immunosuppressive T cell phenotypes, which is helpful because it means that the immune system will be more active for longer. And it will be more effectively active because of the heightened amount of CD8 positive T cells. And it actually, for those T cell types that already are here immunosuppressive, it actually inverts them. To, these cytotoxic ones up here. Two cytotoxics. That's pretty good. So, apparently this polarization of T cells, or this maturation of T cells into suppressive or cytotoxic cells, isn't final, and Cordyceps can influence this and shepherd it towards the cytotoxic side instead of the immunosuppressive side of this maturation polarization paradigm or spectrum. And that is good for us because that means that we have more T cells available that have cytotoxic potential, that have potential in killing cancer cells, which is very, very good for us. And what Cordyceps sinensis also does is that it inverts this M2 macrophage polarization and changes these into M1 macrophages. And M1 macrophages are, I would say, they are eater and destruction macrophages. So M2 are kind of regenerative. They help cells with multiplying. They help with extracellular matrix remodeling, which we will get into in a bit, but right now just shelve that. And M1 macrophages, they eat up destroyed cells, cell debris, extracellular debris, and they eat up bacteria, and they kill sometimes even cancer cells. So they have anti-cancer functionality, and they also promote the maturation of, wouldn't you have guessed it, CD8 positive T cells, so cytotoxic T cells. So Cordyceps sinensis especially, is very, very good at kind of inverting this immune evasive, immune suppressive environment around the cancer tumor so that it is active and that it is cytotoxic. And that is very helpful. And this part here, especially, is very, very helpful for us because a large part of the biomass of a cancer tumor is actually made up of M2 macrophages, which act in this immune suppressive way and act in promoting the proliferation or the growth of the cells of the cancer tumor. If we can invert these M2 macrophages that do this into M1 macrophages, voila, we have now basically an agent that is already behind enemy lines at a large number, that has activity against these enemies and that recruits other agents to the site to kill this enemy. I think that's pretty good, and it gets better. Now, I also mentioned at the start that Cordyceps sinensis has an effect on apoptosis and on cell growth. How does that work out? Well, if we look here at a specific compound that I've already mentioned that is present in Cordyceps sinensis, namely cordycepin. This has various effects. It increases the level of certain proteins. It's not very important what these proteins are, but for those interested, it's caspases, DR3, BAX, and FAS. So if you know what those are, you will know that this increases, I would say it enables easier apoptotic signaling. So this increase in, let's say pro-apoptotic tone, we would say, because it's signaling, but let's just say apoptotic signaling. All right? So these here increase apoptosis, and cordycepin does that, which is good because it kind of, if we go back to the very start, it attacks this part of tumors and cancer tumors, which is good for us. But it also decreases a very well-known protein named BCL2, which is a strong anti-apoptotic signaler. So this means this here basically supports the increase in pro-apoptotic signaling because it decreases anti-apoptotic signaling, which makes it easier for the cell to initiate apoptosis both intrinsically and extrinsically. So now this cancer cell has an easier time killing itself, but also has an easier time to receive death signals from immune cells, which it, for example, does with this FAS. We call it FAS death receptor, or then the ligand that is expressed by immune cells, FAS death ligand. And that just increases apoptotic tone, or increases pro-apoptotic signaling while decreasing anti-apoptotic signaling, which just increases apoptosis or cell death for these cancer tumors, which is very good. Again, what we would want. And what it also does, cordycepin blocks or arrests cells at the cells at G2M phase transition in the cell cycle. Now, the cell cycle is comprised of different phases, and the most important phase in making cells grow, or making cells proliferate, I should say, because they technically grow before that, but is this phase transition here. There's a checkpoint in this transition where the cell goes from G2, which is a kind of growth phase after the cell has reduplicated its genome, and it goes into the M phase, which is the mitosis, which is the actual process of the cell kind of splitting into two cells. And obviously, if we arrest a cancer cell in this, phase transition here, it means this cancer cell cannot replicate. it doesn't split into two cells, which means that the cancer tumor will grow slower. But cordycepin is better than that even. So what cordycepin also does, it decreases the number of cells and the viability of cells that we can observe in G1, or I should write that out, decreases the number and viability of cells at G1 and G2M. This means that we have less cells at this phase transition and less cells at the first growth stage that are actually viable. How do we know this? Well, it also increases the sub G1 population. So increases sub G1 population. What does all of this mean? Well, the sub G1 is basically, it's a, I would say, a dysfunctional G1 cell. So a cell cycle has these different stages. It starts with G1 or with mitosis, depending on how you lay it out. It starts with, let's say it starts with G1, then it goes into S, then it goes into G2, then it goes into M. Now, G1 is kind of the phase where the cell just grows in volume, builds out proteins, multiplies, mitochondria, etc. Then we go into S phase, which is the replication of the genome, of the nuclear genome specifically. And then we go into G2, which is kind of the final growth before the split, the M, the mitosis, the split of cells. And if we have an increased sub G1 population, this means that the step of M was completed unsuccessfully. We now have a population of cells that have not an entire genome, basically. This is what sub G1 means. Sub G1 means it's a G1 cell, so a cell that is in the G1 phase of the cell cycle that doesn't have, the genome that a normal cell in G1 would have. This means it has a defunct genome, which is very oftentimes, an indicator for a pre-apoptotic cell after mitotic failure. This is good for us, because if this happens in cancer, which it does, then cordycepin has the ability to just kill cancer cells that are growing outright, right? And it improves the pro-apoptotic signaling inside these cancer cells, whether that signaling comes from the cancer mitochondria or from outside of the cell in the form of immune signals or immune cell-derived signals. And that's good for us because it means that this down here, specifically, this goes for growing cells, right? If you have kind of cancer cells that are what we call quiescent, so they're not growing, this doesn't really help us. It just stops them from growing, basically, or slows their growth when they do, because their growth is going to be difficult and ineffective. And this up here works against all cancer cells because it just improves apoptotic tone and thus pro-apoptotic signaling, where it's decreasing anti-apoptotic signaling, and this increases cell death, which again, if we go back to this, we now have both of these dimensions up here for the tumor. We have a now increased apoptosis in our cancer tumor and a decreased growth, which of course is good for the patient because it just generally gives the patient more time to live, and especially more time to use other interventions as well to kill the cancer finally, and that is kind of the whole point of doing these interventions, is to give the patient enough time to live long enough for the body to combat the cancer effectively and kill it completely. That's the dream. Whether that's possible in every patient, we don't know, but cordycepin actually helps in this. Now, if you found this interesting so far, please do consider subscribing or liking the video. It helps us out and it kind of tells us what to do more of. And now we get into the other two attributes of Cordyceps sinensis that I talked about at the start, and that is invasion and metastasis. So, um, to understand this, I want to go into why this is important and into a bit of a background or of metastasis, how metastasis works or how we think it works and how it might actually work a bit different, and why that doesn't matter for the efficacy of Cordyceps sinensis. Okay, so usually we think that metastasis is a process in cancer cells which necessitates the cancer cell to run through what we call EMT. EMT is short for epithelial mesenchymal transition, and this means that cancer cells, which largely derive from epithelial tissue, that they go through a transition from an epithelial phenotype to a more mesenchymal phenotype. What does all of that mean? Epithelial is kind of these like rigid cells, like on my skin here, on the eye, on the outside of the eyes, on the inside of my gut, that are very, very organized, very, very, constricted in their growth. They're very, very regulated in that aspect, and they're very, very stationary. Of course, if a cancer remains stationary, how can they metastasize, right? If a cancer cell remains stationary, okay, they will invade, local tissue, but they won't metastasize. So if you have bone cancer, it won't certainly, bone cancer in your leg, it won't certainly be in your skull, on your heart, on your liver, etc. What we see, however, in advanced cancers is that exactly this happens. The cancer will actually spread and will actually metastasize and will actually be mobile, even if it was initially an epithelial cancer. That is, of course, not good. And it does so, or so we have thought, through the epithelial mesenchymal transition. Interestingly, though, new data showed that cancer cells can actually metastasize without this. To me, that was a bit confusing because, if you are a fully epithelial cell or as a cancer cell are epithelioid, you can't really metastasize because you need certain survival signals from your direct environmental cues. So how could the cell, metastasize and be mobile whilst being a cell type that isn't mobile? To me, that didn't make much sense, but it's contradictory to what I understood of cancer. So naturally, I looked deeper into it. I dug into it because how else could I call myself a scientist? Anyway, what I thought here then is that, okay, then we have kind of two explanations of what could happen here, or at least two explanations that made sense to me. One is that for metastasis, we then just see not a transit of single cells in the blood, but of like larger clusters of cells. So we have clustered metastasis, where the cells of the primary tumor won't go out one by one from the primary tumor, but will kind of like bud out as like clumps of cells into the blood, and then travel as these clumps that kind of keep each other alive in or by expressing reciprocal survival signals. That was one hypothesis I came up with to explain this, and then another hypothesis is what I at first thought, okay, we might then just have an EHT, like another type of epithelial to other tissue transition that goes to hematopoietic cells, which are kind of blood cells, and immune cells are hematopoietic cells. And I thought, okay, these are kind of two explanations that make sense to me for how cancer could metastasize without the classical epithelial mesenchymal transition that would make them mobile enough to even metastasize. And it turns out that in literature, actually, yes, this does happen. And yes, this does happen in the form of specifically epithelial myeloid transition transitory cells. Myeloid is a kind of subgroup of hematopoietic cells. So this is a kind of subgroup of blood and immune cells. Specifically, this is like your erythrocytes, so your red blood cells, your white blood cells, your thrombocytes. So it's a specific subtype of your blood and immune cells. And we actually see cancers that use epithelial to myeloid transitions instead of epithelial to mesenchymal transitions. And we also see that generally metastasis is more effective if we have clusters of cells metastasizing than single cells. And we also see that clustered metastasis is indeed bad for cancer patient prognoses. Why am I telling you all of this? If we go back to Cordyceps sinensis, I talk about all of this because it's important to understand this, to understand why Cordyceps sinensis is so powerful in stopping metastasis and invasion across the board for cancer. Now, what Cordyceps sinensis does, or specifically, I should say, cordycepin. So let's look at C. sinensis as a whole, and then look at sinensis, and then look at cordycepin. Which acts at suppressing the expression of what we call matrix metalloproteinase 9. Why is that good? Well, these matrix metalloproteinase 9, or in general matrix metalloproteinases in general, are used for extracellular matrix remodeling. Now, remember, I talked about, or I mentioned this priorly, what this does, ECM remodeling, is it restructures the kind of extracellular scaffolding around the tumor, or generally around cells. This is important because in tumors, or in cancer tumors, for them to metastasize properly, they need to kind of dissolve or restructure, remodel their surrounding extracellular matrix, their surrounding extracellular scaffolding, to actually invade other tissues and metastasize effectively. So by suppressing MMP9 here, matrix metalloproteinase 9, we do suppress this remodeling process to some degree. Now, obviously, there's multiple matrix metalloproteinases, not just the 9er. So, this has limited effectiveness, but it does suppress migration, it does suppress invasion, and it does suppress metastasis, just simply by disallowing or slowing down this remodeling of the extracellular matrix. What Cordyceps sinensis also does, by virtue of polysaccharides, is that it suppresses another matrix metalloproteinase, suppressing MMP1, which does the same, well, not exactly the same thing, but very similar. It also helps in remodeling the extracellular matrix, just as MMP9. And of course, suppressing this is very good for suppressing invasion and metastasis of the cancer. And what these polysaccharides also do is that they suppress VEGF, which is short for vascular endothelial growth factor. And what it does, it stimulates the growth of blood vessels into the tumor, which one supplies the tumor with nutrients, but also gives the tumor access to the blood. We're going to talk about angiogenesis and what it kind of does in cancer development and cancer progression in a future video and future article. So stick around if you're interested for that. But by suppressing this, and by suppressing this, so by suppressing matrix metalloproteinases 1 and 9, and by suppressing VEGF, we significantly slow down the metastatic process by just slowing down the remodeling of the extracellular matrix. So we can kind of picture this as the cancer tumor still being kept inside of a prison, not being able to break that prison down and actually get out through the broken down walls or bars of the prison. And MMPs, help the cancer in remodeling this prison so that there is a door where there was none, so the cancer can escape and metastasize. And VEGF kind of just improves the traffic flow of escape vehicles to the prison. So, the prisoner or the cancer has a better chance of escaping this prison. It's kind of the mental image that this should evoke. And that obviously is very bad for us if we want to stop cancer, if we want to stop cancer from killing people. And it's also interesting because none of these three things here are specific to the mesenchymal, to the epithelial mesenchymal transition, or to the epithelial myeloid transition, or to the clustered metastasis. This helps against all three of those. So it doesn't really matter how the cancer metastasizes, it still needs to break out of this prison, and we can slow that down using Cordyceps sinensis, using cordycepin from Cordyceps sinensis, and using polysaccharides from Cordyceps sinensis. I think, that's a huge, huge thing to have going for us, to have, accessible to us to help cancer patients in their battle against cancer, because it just gives them more time and slows down the aggressive nature of cancer spreading throughout the body. Now, as I've mentioned, I've written an entire article about the prospect of using Cordyceps sinensis against cancer. And you can find on that article all the citations, all the scientific references I used to build this concept out. And also you get a bit more information about the kind of explanations and background of all of this. And you get some extra information on the toxicology, on effective dosing, on clinical trials, and on possible synergism that we can use to improve the performance of Cordyceps sinensis extracts as interventions. And you can find the link to this in the description below. I think it's personally very, very interesting. I think it's very, very helpful to cancer patients, and I would highly recommend you check it out because it just has a bit more information than I can convey in these videos, which are thought to be a bit more approachable. I hope I actually made it more approachable. If not, do comment and call me an idiot. Do so kindly though, and I think with that I will close the video out. If you do want help with navigating the bleeding edge of science with regard to developing specific anti-cancer protocols for you and for your cancer specifically, whether it is interleukin-6 positive mesothelioma or polycythemia vera or whatever it is, you can reach out to us at marchwood.com/outlive and we'll get you situated. We'll get you a specific program, help you along the way of dealing with your cancer. I think that's the best we can do. Obviously, these articles do hold all the information we use, but it's kind of difficult to make specific intervention plans for people I've never talked to, that we've never talked to. And that's why we offer this service. So if you're interested, check it out. If not, no harm, no foul. I wish you swift healing and lasting health, of course. And God bless you.